Okay, so we're at the top of the hour here. We're very pleased today to have doctors Preethi Krishnan and Farooq Syed, joining us from the Department of Pediatrics, Indiana University School of Medicine, to talk with us about the beta cell microRNAs, functioning as molecular hubs of type 1 diabetes pathogenesis and risk. And just a little bio about both of them. As we know, Indiana is a powerhouse of type 1 diabetes research and really bringing so many new researchers, new techniques, new ideas to the forefront. So we're so appreciative that they're joining us today. But for Dr. Preethi Krishnan, she's currently working as a data analyst at Indiana University. She obtained her PhD in medical sciences from the University of Alberta, Canada, where the overall objective of her study was to identify small non-coding RNAs as biomarkers for breast cancer. She moved to the USA and trained as a postdoctoral fellow under Dr. Carmela Evans-Molina. Apart from focusing on biomarker identification, she also learned the molecular techniques needed to delineate the functions of a biomarker. She then moved back to Canada and worked as a postdoctoral researcher in Dr. Jane Hill's lab and identified sex-specific mRNA biomarkers for childhood tuberculosis. Over the past 12 years, she's published about 17 papers as a first and co-author, and her current role involves applying computational and biostatistical methodologies to analyze various high-throughput data sets generated from human samples in vivo, ex vivo, and in vitro model systems. Dr. Syed obtained his PhD in clinical pathophysiology and pharmaceutical sciences from the University of Pisa, Italy. During his doctoral tenure, his research focused on exploring the role of nanomedicine in safeguarding and transplanting human pancreatic islets as a potential therapy for type 1 diabetes. And following the completion of his PhD, he embarked on postdoctoral training at the Indiana University School of Medicine. During his postdoctoral stint, he concentrated on identifying early biomarkers and therapeutic targets aimed at detecting and mitigating stress-induced damage to pancreatic beta cells in context of type 1 diabetes development. And currently, he's engaged in ongoing study at IUSM, delving into the impact of pro-inflammatory cytokines on the regulation of small non-coding RNAs, specifically miRNAs, in vitro models of type 1 diabetes. And drawing on his extensive background in molecular and cell biology, his expertise lies in unraveling the complex interplay between the islet and immune signaling pathways and elucidating how their modulation influences disease progression. And throughout his research, he endeavors to uncover the molecular mechanisms governing miRNA regulation and its profound implications for islet inflammation. His long-term goal is to pioneer innovative strategies for the prevention of type 1 diabetes. And we welcome this dynamic duo to kind of walk us through their newest paper. Thank you both for joining us.
Thank you, Monica. Thank you, Sugar Science, for giving us this opportunity to share some of our recent data on beta cell microRNAs functions as molecular hubs of type 1 diabetes and pathogenesis and risk. Basically, for the next few minutes, myself and my colleague, Dr. Preethi Krishnan, would be doing in two parts. One part, I would take you through the biomarker aspect. The other part, Preethi would be giving the functional relevance of one microRNA, that is microRNA 146, on beta cell function and stress. As known very well for this crowd, that in type 1 diabetes, beta cells are not an innocent victim of immune cell-mediated beta cell destruction. However, they are involved as an active participant in the autoimmune process, whereby environmental insults such as viral infections as well as pro-inflammatory cytokine stress can lead to the activation of endoplasmic reticulum stress, mitochondrial dysfunction, and this leads to the upregulation of MHC class I and upregulation of inflammatory gene signatures and microRNAs and chemokine release and pro-inflammatory cytokine, has been released from these stressed islets. This leads to the presentation of new antigen and the immune cell-mediated beta cell attack on pancreatic beta cells and then development of type 1 diabetes. So given this thing, the major unmet need in type 1D is the identification of specific biomarkers during the early stage of type 1, pre-symptomatic stage of type 1 diabetes, which may offer the opportunity to prevent the ongoing beta cell stress and death in type 1 diabetes. growing evidence suggests that microRNAs, particularly the EV-derived microRNAs, might have potential for the diagnostics of pre-symptomatic cases in the early development of type 1 diabetes. This slide shows you the canonical pathway by which the microRNAs have been generated, and then mechanism by which microRNA control the post-transcriptional gene regulation. The primarily, microRNAs has been transcribed by the RNA polymerase II in the nucleus, and then this has been processed by the DOSHA and GRGC8 complex to form a primary microRNA. These pre-microRNAs can be exported from the nucleus to the cytoplasm by RAN-GTPases and export in pi to the cytoplasm, and then further processed by Dicer1 and TRB complex to form the duplex microRNA. These duplex microRNA has been sequestered by the AGO124 complex to form or anneal the single microRNA to form the mature microRNA. These mature microRNAs can bind to the 3' UTR of any mRNA, which has the complementary seed sequences, and then control the expression of this mRNA either by translation depression or degradation of these mRNAs. This lead to the decrease or increase in certain protein expression during the inflammatory condition. And it's very well known these microRNAs have been highly enriched in extracellular vesicles. From here onwards, I will be calling the EVs as EVs. So basically, EVs are formed by the invagination of early endosomes in the endosomes, and these endosomes can fuse with the multivesicular bodies. Once the multivesicular bodies fuse with the plasma membrane, the cytoplasmic cargo, which has been present in these endosomes, can be fused to the plasma membrane and released into the extracellular space, forming the extracellular vesicles. These extracellular vesicles are 50 to 200 nanometer in size. When they are taken up by any immune cell in the extracellular space, these can activate or suppress the immune cells. Particularly, they have the signatures for cell-specific. So if EV is released from under stress conditions, if they are released from the beta cells, they can be taken up by the immune cells and activate beta cell targeted immune-mediated beta cell destruction. So as
Farooq,
given this,
can I just ask a question on that last slide?
Yep.
What is the concentration of those EVs? Is it variable?
It's we never seen difference between the EV numbers between the pro-inflammatory conditions and the controls. But the packaging of EVs increases. Basically, all the footprints from the cytoplasm can get sequestered into these EVs and then released into circulation. So the amount you get around 10-fold increase in the content, but not in the numbers. Yeah.
Interesting. Okay. So more goods in the box, but same number of boxes.
Yes, 100%. As given this background, this slide shows you the outline of my today's presentation, where we try to ask the question, what are the impacts of pro-inflammatory cytokine on microRNA regulation in human pancreatic islets? And then how does the islet microRNA and the EV microRNAs, is there any commonality and differences between the islet-derived and EV-derived microRNAs? And whether we could be able to utilize these microRNAs in the EVs as a biomarker for the detection of yearly beta cell stress and death during the evolution of type 1 diabetes. And then what are the therapeutic potential of these microRNAs in preventing of beta cell stress and death in preclinical models of type 1 diabetes? In part 2, my colleague, Dr. Preethi Krishnan, would be presenting the role of microRNA 146 in evolution of type 1D pathogenesis. And then what are the mechanisms that have been involved by regulated through the microRNA 146 during beta cell stress and dysfunction. So as said, in the part, this slide shows you the overall project workflow of my part 1, where human islets have been treated with pro-inflammatory cytokines, particularly interferon alpha and interferon gamma and IL-1 beta. And conditioned media from the same islets have been collected and EVs have been isolated. And we subjected both islets and islet-derived EVs for small RNA sequencing, particularly microRNA sequencing, performed a bioinformatic analysis to identify the differentially expressed microRNAs, and then used multiple platforms to validate our identified differentially expressed microRNAs. And then we utilized our main goal is to see the clinical utility of these microRNAs in detecting yearly type 1D as well as ongoing beta cell stress or death. So we isolated EVs from plasma samples of new onset type 1 diabetic autoantibody positive individuals as well as healthy controls to see the utility of this microRNAs in predicting ongoing beta cell stress and death during type 1 diabetes. This slide shows you our human islet-derived microRNAs that have been identified through our small RNA sequencing. In total,
just pausing. Oh, sorry, just pausing. I have a question. Which types of pathways are MIR 146A5P dysregulated? This is coming from Bettina Pedersen.
It's basically, in our case, we identified mitochondrial dysfunction is one of the main, when microRNA 146 upregulates, it shuttles to the mitochondria and then, dysregulate the mitochondrial function, leading to mitochondrial dysfunction and death in beta cells. My colleague Preethi would be explaining all those things that we have identified in this study.
Great, thank you.
Out of these 2,740 microRNAs, after filtering for read counts, we could able to retain 1,110 microRNAs, out of which 20 microRNAs have shown differential expression pattern. Out of the 20 microRNAs, 15 microRNAs are upregulated in cytokine treatment condition and 5 are downregulated. Further, to validate these differentially expressed microRNAs, we obtained a separate batch of human islets and treated with pro-inflammatory cytokines, and then performed RT-PCR analysis to see the validity of our small RNA sequencing. We could able to identify 95% of these microRNAs showed similar trend. However, only 50% of microRNAs showed statistical significance through our qPCR analysis. Next, the question is whether the EVs that I was talking about before, whether we could able to identify pure EV fractions. So to do that, we isolated EVs from the conditioned media from the human islets and then subjected them for immunoblot analysis. As shown over here, two tetraspanin markers, CD63 and CD9, has been enriched in islet-derived EV fractions, implying like the purity of our EVs. And we also performed the interferometry analysis. Basically, these are ExoView based analysis where in silica chip, you print the antibodies for CD63, CD81, and CD9. And then you can incubate your supernatant or plasma samples so that the captured antibody captures your EVs depending upon their surface markers. And then by using a detection antibody, you can identify the fraction of EVs that contain different tetraspanin markers. As shown over here, from our analysis, we identified CD63 and CD81, both in control as well as cytokine treated EV conditioned media have been enriched in islet-derived fractions, implying like these two markers might be highly expressed in the EVs of islet-derived EVs. Next, we asked to characterize further the size of these EVs that have been isolated by our approach. We performed nanoparticle tracking analysis, and we identified that EVs are around 50 to 200 nanometer in size, a median size of 100 nanometer. And as you can see here, to our surprise, we haven't observed any difference in particle numbers of the EVs between untreated and cytokine-treated samples. Next, we asked whether these pro-inflammatory cytokines can also activate the EV biogenesis pathway. We looked into a few genes that are involved in different steps of EV biogenesis, the TP53, RAP27A, and RAP27B. are the three markers that we looked from the human islets. As you can see, there is a significant upregulation of these EV biogenesis markers in islets treated with pro-inflammatory cytokines. Next, we subjected the human islets that have been untreated and cytokine treated with the scanning electron microscopy and transverse electron microscopy. As one can clearly see the presence of the EVs in the plasma membranes of the human islets, both in the control and cytokine-treated human islets. We also observed by the transverse electron microscopy the presence of EVs in the plasma over the plasma membrane as well as in the cytoplasm, showing multivesicular body, which has been represented by the arrow.
Farooq, when you're looking at these islets, are you able to distinguish whether it's an alpha or a beta cell that you're seeing these changes occurring in other cell types within the islet?
Yep. Yeah, we can see that one by using the transverse electron microscopy. You can see your beta cells containing the insulin granules.
Yep. Yeah. And how about the other ones? Alpha, beta, alpha, delta, etc.
We haven't looked, that's a very good question, but we haven't looked into the other cell type since we are more interested in how does beta cell stress could lead to, the activation of this. That might be one thing that we can go back and look into those, other cell types too.
Thanks.
So we isolated the EVs and then further characterized their structure, ultrastructure, by using the scanning electron microscopy and then transverse electron microscopy. Both of these approach confirm that the EVs have a similar size that has been, previously published in literature, showing that our isolation protocol could able to enrich for EVs that are with the, similar shape and structure. Then after confirming our EV characterization, we isolated RNA from the EV-derived EVs and then subjected them for small RNA sequencing. As shown over here, we identified around 890 microRNAs from the EVs and 14 microRNAs that showed differential expression pattern. Out of the 14, 11 microRNAs are upregulated and 3 microRNAs have been downregulated in the EV fractions of the islets treated with pro-inflammatory cytokine. We, similar to our islet validation studies, we performed qPCR analysis on this islet-derived EV microRNAs and then identified 5 microRNAs that showed significant difference among control and cytokine-treated conditions. But our goal is to identify a composite of microRNA signatures that could able to reflect the ongoing beta cell stress and death. So in collaboration with the bioinformatics group here in IU, we utilize machine learning approaches to identify and the differentially expressed microRNAs that might have potential, therapeutic diagnostic potential in ongoing beta cell stress. So we performed the decision tree, learning vector quantification, random forests, and support vector machine learning algorithms and ranked them from 1 to 10. Out of this top 10 ranking, we picked 5 microRNAs that has some relevance for beta cell stress, TLR activation, cell-to-cell communication, mitochondrial dysfunction, or ER stress activation. These are the 5 microRNAs that showed these characteristics and also involved in insulin secretion in beta, some way it is regulating insulin secretion, involved in insulin secretion mechanisms. So qPCR and other approaches are really cumbersome. You have to isolate and, that might take a day for perform an experiment. Our goal is to make from bench to bedside. So in collaboration with Dr. Rajesh Sardar and Sarah is graduate student from IU School of Science, where they have developed LSPR, which has a surface plasma resonance biosensor approach, where this approach utilizes triangular nanoprisms, which have been functionalized with then Spreon hexoethanol, particle. And then these functionalized particles, once they have been UV irradiated, the opening of this ring could lead to the zeta ionic functionalization that leads to the binding of your antisense, either single stranded DNA or antibody. You can utilize for multiple approach. In our case, we have used single stranded DNA as an antisense for your microRNA of interest. And this, once it binds, you can, by using the delta LSPR, you can use the plate reader to identify the difference between the closed ring as well as the once you are functionalized single stranded the oligonucleotide bind to this one. You can find the differences between using a plate reader. Once you just incubate your supernatant with this uh, uh, functionalized composite, and once your microRNA bind to that, there is a difference in the OD of this wavelength. The difference between your control, that is the positive control, which has the only with your single stranded DNA, and then once the microRNA binds, the difference can be utilized to identify the concentration of this microRNA in a given solution.
Farooq, what is the volume of blood that you need to run that pipeline?
This is just 25 microliter is enough.
Oh, great. Yeah, I wondered also, this is kind of this might be a question for later, but do you imagine that patients, might display this kind of a microRNA signature across endotypes?
Might be, yeah. So we tested in, we haven't tested in endotypes, but we tested in type 1D and autoantibody positive individuals. And interestingly, I have some results to show you now. We've seen some really significant differences
Okay.
when compared
great.
to healthy controls. Yep.
Thank you.
So to validate this approach, we used a separate batch of human islets and isolated EV from that conditioned media from the control and cytokine treated conditioned media, and then subjected them to the LSPR approach. As shown over here, similar to our qPCR analysis, we have seen a significant difference in the microRNA expression pattern of all these 5 microRNAs that we have identified, implying this approach could be able to differentiate between cytokine and untreated condition and the enrichment of microRNAs in these EVs. Here, as Monica asked, we have Indiana. We have a human biobank of samples from controls, autoantibody positive individuals, as well as type 1D donors. We isolated EVs from the plasma of these donors and then subjected them to this LSPR approach. As shown over here, all these 5 microRNAs similar to the EV derived microRNAs from islets, they have shown the similar pattern of expression, implying that what we have mimicked in the in vitro model for the type 1D could be able to recapitulate these things in the in vivo settings of human donors samples. Very interestingly, one thing we observed was 3 microRNAs, 146A, 5P, and 802, and 124-3P was shown significant difference in autoantibody positive individuals, implying maybe this 3 microRNAs can be utilized to identify their beta cell stress status in autoantibody positive individuals, whether one could be able to develop or progress to type 1 diabetes or not. By using these composite microRNA signatures. But our question is, so to identify common microRNAs between the islet and islet-derived EVs. So we performed bioinformatic analysis to identify the common microRNA between both islet and islet-derived EVs, as well as in literature, as well as we identified, there is a difference between male and female microRNA signatures, because microRNAs have a sexual dimorphic pattern of expression. To make sure our biomarkers could have some potential for both male and female, we performed bioinformatic analysis and identified 2 microRNAs which had a common regulatory pattern between islet and islet-derived EVs. So as shown here in the bar graphs, microRNA 155 and microRNA 146 both showed similar trend and similar pattern between male and female donors. between control and cytokine-treated islets, implying these microRNAs can be further validated for their potential therapeutic applications and their function in beta cell, stress condition. So to see whether whatever we identified from islets, because these are all crude lysates, and then we don't know whether they are coming from beta cells or not. So we received pancreatic tissue sections from the NPOD consortium, and then subjected them for single molecular fish analysis using RNA-scope approach, and then seen the expression pattern of this microRNA, particularly in this case, as a proof of concept, we performed for microRNA 155 to see the expression pattern. As one could able to see and appreciate the expression pattern of these microRNAs in these human pancreatic tissue sections present mostly in the insulin positive cells. It's really encouraging for us to see this are upregulated in type 1D as well as in the autoantibody positive individuals. In collaboration with Dr. Jing Liu in School of Science and his student, PhD student Gary Chang, we further want to see the expression pattern of this microRNA in these pancreatic tissue sections. So the spatial expression pattern showed that microRNA 155 has been upregulated in type 1D and autoantibody positive individuals, which is reflecting what we have seen in our in vitro settings, as well as the spatial expression pattern of this microRNA between cytoplasmic to nuclear ratio has also been increased in type 1D and autoantibody donors, implying there is a spatial differential expression pattern of this microRNA under stress conditions.
There was a just question, Farooq, from
Yep.
Dr. Mirmira, wondering,
Sure.
how do you know that the circulating EV microRNAs are coming from islets and not other circulating cells?
So this is a very good question, because we haven't had any specific markers for islet-derived EVs currently, but we are trying to identify these surface markers that are only present in EVs. But with the current approach, only with the microRNA signatures, we can tell with our in vitro model, we could able to see similar expression pattern in the plasma EVs. So that is the only thing we can currently tell. We are trying to identify islet-derived EV specific signatures for EVs. Maybe in coming months, we might have some data out of it to see one could able to clearly enrich for islet-derived EVs during the early and late phase of type 1D evolution. Does that answer his question?
I think so. Yes,
Okay.
he said yes.
So to further see the potential of this microRNA in, identifying the beta cell stress in the type 1D and autoantibody positive individuals, we utilize the machine learning algorithm, particularly random forest classification approach, to see what kind of expression pattern could imply the stressed beta cells when compared to healthy controls. So around 124 different classifiers have been utilized in this machine learning algorithms. Out of this, 4 classifiers showed like really higher ability to predict the stressed beta cells, particularly the clustering effect of these RNAs and their clustering near the nuclear membrane, and then distribution in the cytoplasm, like single molecule distribution in the cytoplasm, and as well as the boundary expression in the boundary of this microRNAs. So all these 4 classifications, when you apply, we could able to clearly identify, this could able to tell in autoantibody positive individuals as well as type 1D donors, there is an stress. And this spatial distribution could able to reflect that ongoing stress of this microRNA distribution in this beta cells. Finally, the question is like whether this microRNAs have any therapeutic potential. In collaboration with my longtime friend and collaborator, Dr. Kentaro Hamada, we generated AAVs, which have been, the antisense of this 155 has been cloned, which has been driven by the RIP promoter to have beta cell specific, by expression pattern. We injected the female NOD mice, 4 weeks of age, and then subjected them for the GTT, IPGTT. As shown over here, you could clearly see there is a significant difference in the mice that have been treated with the microRNA 155 5P compared to the scrambled and PBS treated mice. And this also been clearly implicated over here in the area under the curve. There is a significant difference in where this knocking down of this microRNA 155 could able to tightly regulate the glucose control and have better insulin secretion.
Farooq,
Currently,
is there any other human disease where microRNA knockdown is used clinically?
There is one ongoing clinical study, I think, where they utilized a signature of 5 microRNA in gene therapy approach to target some other cancer models, but not in autoimmune diseases.
Yeah.
Yep. But this is the initial thing. Once we have some preliminary data, we can think of either to deliver these microRNA using lipid nanoparticles or AAV approach to do tissue-specific gene therapy.
No, it's excellent. You're making great strides.
Yeah. So currently we are performing the insulitis from these mice, which have been treated with AAV to see whether we could also, reduce the insulitis in these mice. Maybe in coming weeks, we might have results for this one, to show whether there is an effect of this knocking down on prevention of infiltration, immune cell infiltration in pancreatic tissue sections. So far, in summary of my part 1, I have showed you that EVs isolated from controls, cytokine-treated islets, do not show difference in size or number or morphological or structural characteristic differences. And we observed that significant difference in microRNA cargo loading into the EVs. A significant difference in microRNA expression pattern between cytokine-treated islets and islet-derived EVs has been observed. Although I haven't showed some of the data where we also observed a sexual dimorphic pattern of expression of this microRNA between male and female donors. One advantage of our data is we have 5 male and 5 female out of these 10 donors. So we could able to clearly see the difference. And we also identified a signature of microRNA that might reflect somehow ongoing beta cell stress and death in both in vitro and in vivo clinical cohort patients. And the spatial expression of microRNA from NPOD donors implicated a distinct pattern of microRNA 155 expression in human pancreatic tissue sections of autoantibody positive individuals and type 1D donors. Next, my colleague, Dr. Preethi Krishnan, will take you through the role of microRNA 146 in inflammation and mitochondrial dysfunction and apoptosis. Preethi?
Before Preethi begins, there's just a question from the chat from Bettina Pedersen. Where were the cohorts from again?
This is from our in-house cohort. We have in Indiana for past 5 to 7 years, we are collecting, blood samples from new onset who are within 48 hours of diagnostics. We collect blood samples from these kids, as well as the pathway to prevention trial. We have some samples for the autoantibody positive individuals, and we have healthy controls from our outpatient clinics. We have biobank here.
Yes. And then from Leah Gomez Munez in Spain, sorry, maybe I missed it. How did you stain the MIR 155 in pancreatic sections?
So we used in situ single molecular FISH analysis. Basically, 2 years back, we can do only for pre-microRNAs because the size of microRNA is 22 nucleotide in size. It's very difficult to design probes to identify these microRNAs. But currently there are improved methods from RNA scopes. They have brought to identify mature microRNAs. Currently, I am performing some of the experiments to see what we have seen with the pre-microRNAs could also be, we could be able to see the same expression pattern with the mature microRNA.
Great. On to Preethi.
Thank you, Farooq, and thank you, Sugar Science team, for giving us the opportunity to present today. Like Farooq mentioned, so I would be running you through some of the findings that we have from our recently published paper on 146.5p. Yeah. Okay. So what do we know about this microRNA so far from our previous publication? Like Farooq talked in detail, but just a brief out on what we saw. So 146 is highly expressed in islets, islet-derived extracellular vesicles treated with cytokines. And then even in patient samples, we see a higher expression of 146 in T1D when compared to healthy controls. So apart from this, we also have seen that 146 is associated with beta cell dysfunction and death. So one of the papers that talks about the function of 146 is that when 146 is expressed in higher copies, then it leads to the higher cell death. And then if you treat it with an inhibitor, even in the presence of cytokines, the number of dead cells reduces. So which means that 146 may have a role in beta cell dysfunction and also the beta cell death. So so far, yes, this is like a summary. So beta cells, when it undergoes inflammatory stress, it undergoes beta cell death. And then there is a higher copy of 146A5P. And there is a link that is established between higher expression of 146 and beta cell death. But how does this lead to the death is something that is still unclear. And we are trying to understand what kind of mechanisms that 146 can be involved that leads to a dysfunction or the death of a beta cell. So to understand that, first we checked the expression of 146 in different models of type 1D, like we took mouse islets as well as we took MIN6 cells and treated with cytokines. And then in both islets as well as MIN6 cells, when there is an inflammatory stress, we saw higher expression of 146A5P. And then to further understand what exactly it does, we developed stable cells using lentiviral vectors. We have a control cell, which was transduced with scramble sequence. And then we also inhibited the expression of 146 and we overexpressed the expression of 146. And we subjected all of these cells to cytokine treatment. And we, to check if there is apoptosis happening, we did a real-time cell death assessment assay using InQSight system. And here is a representative result from the experiment conducted. The upper panel here is the results from the cells which were inhibited with 146. And as you can see from these graphs, the apoptosis reduces when there is an inhibition of 146, even in the presence of cytokines. Whereas in the lower panel, where we present results from the overexpressing cells, in the effect of microRNA overexpression, especially under the cytokine stress, the number of dead cells is really high. So which again establishes a link between 146 and cell death. And then this confirms what is already known about 146. So to further understand what are the overall mechanisms that it may have an effect on, we performed RNA sequencing. And we looked at the different pathways that gets affected because of different conditions, like what happens when the 146 is inhibited, what happens when it is increased, and what happens when there is a cytokine influence on both the overexpressed as well as inhibited cells. So these are some of the pathways that we have, but I want to draw your attention to some of the pathways that we are focusing today. So that is we saw a rich enrichment of mitochondria-related pathways, and so we hypothesized that 146 might be involved in mitochondrial functions, and maybe that is one of the mechanisms through which the beta cell can be dysfunctioned when there is an overexpression of 146. So we also know from literature that 146 is classified as a mitomer, which means it is present in mitochondria, and then it can influence the effect, or it can influence the function of mitochondria by being present in mitochondria. And so we first wanted to check what happens in Wnt6 cells. So we did subcellular fractionation and then dropped it digital PCR to check if this microRNAs is expressed in different fractions of a cell. So we only took cytoplasm and mitochondria, and in the cytokine influence or under cytokine stress, there is higher copies of 146 in both cytoplasm as well as in mitochondria. So, so once this was established, we thought maybe 146 has an effect on mitochondria function. So how do we go about testing that? So first, we looked at the metabolic function using Seahorse assay, and then we also did electron microscopy to understand if there are ultrastructural changes, and then we also imaged for certain mitochondrial function markers. So these are some of the results from the Seahorse assay, which calculated the oxygen consumption rates under different conditions. So these are some of the parameters represented from the results that we got. And here, as you can see, the basal respiration rate as well as the maximal respiration rate, the cells which were inhibited with 146, the oxygen consumption rates were higher. And when you go to the ATP production rate, I mean, the oxygen consumption rate happening during ATP production, the consumption rate was really less was significantly lower in cells which were overexpressed and treated with cytokines. So this clearly suggests that there is an effect on mitochondrial function when there is an overexpression of microRNA 146. And next, we checked what happens to the copy numbers using ND1 as a marker. So we checked the number of copies that are present when the cells are inhibited with 146 and when they're overexpressed. So there is a higher copy number of ND1 in ZIP cells. And when you look at the ultrastructural images, here the top panel here, these are the cells which were not treated with cytokines, and the bottom panel here has cells which were treated with cytokines. If you look at the cells which are inhibited with inhibitor, in both the presence and in the absence of cytokines, we see a higher copy number of or higher number of mitochondria, but this phenomenon was not seen in cells which are overexpressing microRNA 146. We also see some of the other ultrastructural changes when there is an overexpression of 146. For example, we see an elongated endoplasmic reticulum here. So these are some of the observations from our electron microscopy experiment. And next objective was to further confirm if there is mitochondrial dysfunction happening or not when there is an overexpression of 146. So we used a marker, TIM23, which is an inner membrane protein. So when there is a lower expression of TIM23, it is suggested that there is a mitochondrial dysfunction happening in the cells. So we looked at the expression of TIM23 in all three groups of cells, both in the presence and in the absence of cytokines. And here, if you look at the quantification, so there is a clear downregulation of TIM23 expression when 146 is overexpressed, both in the presence and in the absence of cytokines. So this again confirms that there is a possible dysfunction of mitochondria happening when there is an overexpression of 146. So these are some of the results from our in vitro findings. And then we also wanted to check what happens to mitochondria function in vivo models. So for this, we took NOD islets and isolated islets from different time points. And we did staining for the mitochondrial function markers. And we also did PCR to look at the expression of 146. So first, when you look at the expression of 146A5B, as the disease progressed, we saw higher expression of 146. And if you look at the expression of the mitochondrial function markers, TIM23 and TOM20, there is a gradual decrease of these expression markers along with the overexpression of 146. So this inverse relationship between 146 and the mitochondrial function markers may suggest that the 146 is associated with mitochondrial function. So these are some of the findings that we have from our 146 study. To summarize everything and to put the findings in a nutshell, so when there is an overexpression of 146, we see a higher mitochondrial dysfunction, or number of dead cells are increasing, there is a lower number of mitochondrial copies, and then the ATP is also reduced, and there is a reduced respiration rate. But when there is an inhibition of microRNA 146, even under the influence of cytokines, the number of dead cells is lesser, and there is no mitochondrial dysfunction, or there is lesser amount of mitochondrial dysfunction, and there is a reverse effect that's happening again compared to what was seen when the 146 was overexpressed. So yeah, so these are some of the findings that we wanted to talk today, and we would like to acknowledge the entire group that was involved in conducting this study. So especially even Smully in our lab and all the collaborators, and also the funding agencies and all the donors without whom these studies would not have been possible. Thank you for your attention, and we would be happy to take any more questions if you have.
Thank you so much for that quick tour, but an excellent one. Really exciting work you have, you both and your team are doing at Indiana. I did want to ask you just a couple questions and open it up to the audience to drop questions into the chat or unmute themselves. I wondered, this is sort of a bigger question, but you know, given that the road to stage 3 diagnosis with type 1 diabetes is sort of a relapsing and remitting disease, or at least that's the hypothesis. Do you anticipate that the microRNA signature, the 146A5P signature, will persist across that trajectory, or would there be an optimal window to capture those changes? What do you just, I mean, obviously you don't have the data yet, but I mean, do you have any thoughts?
I think the 146 is very interesting. If you think about 146, just upregulation alone doesn't induce basically the beta cell death. Only when you add cytokines, because it's like a sensitizing thing, you know, marker or factor, which sensitizes the cell for even with a small inflammatory signal, the cells can cook. Maybe, you know, we were thinking like maybe 146 might be a good tool for cancer models where you can overexpress 146 in the cancer tissues and then, you know, kill the thing. But in type 1D, from our data, we can say, because these are 48 hours after the whatever we looked from the donors, right? Within 48 hours of diagnostics. But if you look into the end part tissue sections, they are higher duration samples. Whenever you have basically the presence of insulin positive cells, we could able to see increased expression of this 146 in insulin positive cells. Meaning, if you have residual beta cell, there would be possibility that these cells are stressed out and 146 is up. This might reflect in your circulation.
Yeah, I'm just curious, like how will not only just only for 146A5P, but sort of like the whole panel, you know, of the signature of microRNAs. How might that be used to really capture these, you know, the progression during the prodrome?
Yeah, that could be a good thing, but unfortunately, we couldn't able to do that one with the because we don't have any prospective study samples to follow up on this, you know, how this signature might change, or does it able to, you know, reflect the ongoing beta cell stress or not? Since we have all the cohorts, cross-sectional cohorts, it's very difficult. But one thing we can do is we can try it with the mouse models to see in NOD with different during the progression to diabetes. We can look into these things and then see whether does it have the same, you know, expression pattern or not.
We have Leah Gomez Munez in the audience. We hosted her a while back, and she spoke about the microRNA 30D5P that modulates the PD-1 expression and Treg expansion. I don't know if she wanted to comment or ask a question about how her work may or may not intersect here. Oh, not sure if she's still here. We'll see. But it is kind of an interesting question, you know, how all these, you know, microRNAs may intersect. Here's a question from Andrew Taylor. Did you look into complex 5 activity specifically? That's in the mitochondria. Or do you anticipate that the change in ATP respiration is due to something upstream of the electron transport chain?
Preethi, or should I?
I mean, we haven't looked specifically into complex 5 activity, but yeah,
The Western blot.
yep.
So from the Western blot, I can tell like we have seen there is a decrease in complex 1 if the Western blot, right, Preethi? The expression of complex 1 has been reduced.
Mm-hmm.
The OXPOS,
Yep.
Western blot, yeah. Not we haven't looked into complex 5 separately.
Okay, yeah, that's interesting. And then this is sort of a really a big question. Okay, wait. MIR 146A and MIR 146B are known to be induced in endothelial cells upon exposure to pro-inflammatory cytokines. Could this be an extrinsic effect on beta cells in T1D? That's from Shamik Banerjee.
Yep, there are possibilities. So as far as you have some environmental insults, automatically it's like a stress response thing, I feel. It get activated. Maybe it is to slow down your ATP generation so that cells can recover back. So that's how we think, but we need to further characterize this one to see the relevance of this microRNAs in broader spectrum.
I would also wonder, have you spoken more specifically with Anil Bhushan and Peter Thompson regarding their work in senescence?
Yep.
And how you're finding that, sort of mitochondria are going offline, how those hypotheses and findings could play together.
Yep. I have talked with Peter Thompson in NPOD this year, but I haven't talked about these things. But one thing we know from Kamala last week, she has given a talk in Harvard, and then someone has asked the question like 155 induced senescence. So that makes sense, but yearly senescence, if you think about Pfizer agreeing, a recent cell metabolism paper, maybe yearly senescence is needed for the cells to, prevent the autoimmune mediated beta cell destruction. But chronic activation of these markers might induce apoptosis. That's how I feel like after seeing that paper, maybe this has something to do with the, 155 is shown to be involved in senescence. So I don't know about other microRNAs, but I know like 155 is involved in senescence.
Great, thank you. Alberto Palesi from City of Hope is ready to ask a question.
Sure.
Yeah, I had a couple of questions. One was for
Alberto.
Preethi. She showed her work with the mitochondria and so forth. Did you look in the cells for changes in what genes might be affected by overexpression of 146A? The other thing is that both 155 and 146A are known to be important in T cells, including Tregs for 146A. So at least in the NAD mouse, have you had a chance of looking into those? If there were effects or if you overexpress, is it also going to be expressed in T cells or what if you're looking at?
Yes, we did look at the expression of some of the genes which are acting as targets of 146. So we focused particularly on genes which are involved in insulin secretion, apoptosis, and also the mitochondrial functions. And one example is GPX1, which is involved in mitochondrial functions. And then, so the GPX1 expression goes high when the cells are inhibited with 146, but its expression goes down when the cells are overexpressed with 146. So that is one of the targets that I can think of, and the genes which are probably involved in the functions too.
The other question for Alberto, I think Alberto, it's a very good question. So we haven't looked into the immune part yet, but I think that is something to look for. What are the functions in immune cells, right? Like if you think about Francesco Detta's that cell metabolism paper with Robert Maloney showing like 155 is also overexpressed in T cells, activated T cells. So maybe something to look for what would be the relevance of their function there, whether they involved in activation of the T cells or whether they are, inducing apoptosis in immune cells too. We don't know that one. Maybe something to look for. Yeah.
Great. Just a couple, like two more questions here. MicroRNAs, some of them, including a microRNA 146 and 155, are commonly affected during bacterial infection. So, you guys, basically sprinkled on the cytokines, and that's fantastic. I wonder if you could back up and hypothesize about bacterial versus sort of the CVB viral insult, and whether you've seen anything that might differentiate the responses to either a bacterial or a viral infection.
Unfortunately, I can tell you one thing from my collaboration with sepsis studies. When you do LPS treatment, the bacterial infection induce similar antiviral response. They activate genes, all the antiviral response genes. So any inflammatory or infections might induce these genes involved in antiviral response elements. So you might not, it might be very difficult to differentiate between a bacterial infection comparing to EBV or, a coccidiovirus infection. So that might be difficult to dissect, but there would be certain genes that might be like particularly upregulated. For example, if you take these GBBs, all the GBBs are antiviral response GTPases, which get activated only with type 1 or type 2 interferons. But I don't know whether these GBPs are also upregulated or not in the bacterial infection thing. Maybe those things could able to dissect which infection could have, activate what kind of pathways. Yeah.
Yeah, it's very interesting kind of line of questioning. And then, if we go back to, right, we were talking offline a little bit beforehand, in 2015, Francesco Dauda and company had a great, coming out of University of Siena, had a great review when they were talking just about microRNAs as novel players in the dysfunction between pancreatic islets and the immune system in type 1. And he really did kind of address both sides of the equations. He basically said, obviously in islets, including the beta cells, microRNAs are involved in the cell differentiation as well as insulin secretion. In immune cells, microRNAs have pivotal roles in development, activation, and response to antigens. So he really looked at both sides of the algebraic equation there. And just sort of like, if you could extrapolate, do you think microRNAs could be the best chance for kind of seeing both sides of what's happening in type 1 diabetes in a biomarker sense?
Yeah, for sure. Because if you think about just microRNAs by itself, right? The stability in circulation and then the package into the extracellular vesicle, and then extracellular vesicles, act as a cell-to-cell communication tool for between different organs and different cell types to send signal, whether danger or secretory pattern. Maybe, yeah, they might have some, balancing act between both the compartments. But the thing is, what we are trying to do now is to see the relevance. One thing is whether you isolate the EVs from the pre-diabetic NOD mice and then inject them into the BL6 to see if we have inflammatory, signatures present in these EVs, could they be able to induce immune activation and then lead to like immune cell infiltration in the model where you might never see an immune cell infiltration, right? And the same thing, if you isolate an EV from the BL6 and then do an, transfer into NOD mice and then see whether they could able to prevent the onset of diabetes or delay the onset of diabetes. Those questions might, give us whether you can use EVs one way as a treatment therapeutic for, from a healthy control and then do the transfer. The other thing is what are the signatures they present in those EVs that might have this kind of beneficial as well as, detrimental effect on beta cells, stress or death. Those are the things we are trying now, because we have a model, a mouse model, where we have the reporter CD63 expressed in the pancreas, beta cell specific. So we can clearly enrich this from the plasma and then try to do the islet derived beta cell derived EV adaptive transfer to see their effect. And then you can do sequencing or proteomics to see what kind of signatures present in this EVs.
Yeah. Sort of like it's kind of interesting horizon to move towards, really. Okay, last call for questions. This was such a popular talk and well-attended talk, and I think a lot of people have some great takeaways from it. So thank you both very much for walking us through the data, and we look forward to seeing what comes out of your laboratory next.
Thank you, Monica.
Thank you,
the team, for giving us this opportunity.
Thank you so much for sharing. We'll talk to you again. Bye-bye.
Bye. Take care.